Welcome friends, welcome to the biology classes. Today we will discuss a new chapter, that is reproduction in flowering plants. That is angiospermic flowering plants, how they reproduce. So reproduction are of two types, asexual and sexual reproduction. This pertains to sexual reproduction. As you know that the flower has four parts. From class 8 you have started that flower is made up of calyx, corolla, androecium and gynoecium. These are the four parts of a flower. Flower, do you know that why flower is called a modified shoot? Let us discuss that one first. Suppose this is a plant. Here it is internode. From internode, branches are produced. So these are called internode and this is called node. Here, so flower can be defined as a modified shoot. How? Suppose in flower, here it is calyx. Calyx are green in color and they encircles the entire stem. Calyx. Then comes the corolla. Corolla is a colored one. It is green in color or corolla is red in color. It also encloses the entire stem. Calyx, corolla. Then we start further. Then you will find here androecium. Androecium. If you move further, we will see here gynoecium. Calyx, corolla, red in color. This is usually green in color. As in hibiscus rosa sinensis, you have seen this is calyx and corolla is red in color. Then these are our androecium and gynoecium. Now these are the internode and these are the nodes. Now if you press the stem in this way, what you will find? You will find that this is called thalamus. In the thalamus, these four holes, these are called holes. These four holes are there and they are suppressed. So if it gets suppressed, the first calyx will come. So if you cut it, you will see calyx are here. Then you will find corolla. Corolla is here. Calyx, corolla. Then you will find androecium here with filament and anther. Then you will find gynoecium. So these are internodes, these are internodes, these are internodes. So internode and node are so suppressed that you cannot distinguish. But if you extend it, you will find this. So these are the four holes. Now we know that calyx is made up of, this calyx is made up of, we say, sepals. Corolla is made up of individual member are known as petal. Sepal petal. Androecium, individual are known as stamen. And gynoecium, ovary style stigma, these are called carpets. These are called carpets. So many sepal makes a calyx. So many petal makes a corolla. Stamen is anther and filament. This is filament and here is anther. Stamen. And carpels are made up of ovary, this is style, and this is stigma. So carpels are made up of ovary, style, or stigma, stamen, filament, and anther, petal, and sepal. These are accessory holes, not required for reproduction, but they protect the Androecium and gynoecium on the outer side. And these are called essential holes. These are known as essential holes, WHORLS holes. These are called accessory holes. So these are the four parts of a flower. Now we discuss these two for the purpose of reproduction. Clear? And this is the thalamus on which this flower grows. Now let us start with the Androecium. Androecium is made up of filament and anther. So this is filament and anther remains like this. Filament and anther. Okay. Now we'll cut it. We'll cut the anther, not filament. Filament is holding it. It can hold dorsal from the dorsal side called dorsifex. It can hold from here called basifex. So many other nomenclatures are there. So we'll cut it. If we cut it, we will see made up of two lobes. And these two lobes the outer one is epidermis. This is called epidermis. In the initial stage, if you cut the anther, it will see that it contains a mass of meristematic cells. Mass of meristematic cells. So there are the anther initially looks mass of meristematic cells in which and we are producing male gamete, or before the male gamete we are producing microspores. These are also called pollen grains. So microspores or pollen grains, they are developed in the anther. In the anther, we see initially there are two lobes, bilobe, and this is full of meristematic cells. What is meristematic cells? Meristematic cells are the cells which divide rapidly. Once they divide, go on dividing, they become stagnant and they become permanent cells. So meristematic cells, they are in the active growth. So in the active phase of growth, the meristematic cells. So these meristematic cells ultimately will form the microspore or pollen grains. Now, as the anther grows, it becomes four-lobed. It becomes four-lobed. The two-lobe structure, as the anther grows, it becomes four-lobed, surrounded by this epidermis. Surrounded by epidermis, and the cell grows here. The cell grows here. These cells are called epidermal, and these cells will be called hypodermal cells. In hypodermal cells, some of the cells becomes dense. And they are seen to contain distinct nucleus. So some of the hypodermal cells of the anther gets dense and their nucleus becomes distinct. So these cells are called archisporial cells. Clear? Now these cells are called archisporial cells. Out of the meristematic cells, some cells, the contents of some cells become denser and they become prominent and the nucleus becomes distinct. So these cells are called archisporial cells. Clear? Next. These archisporial cells then divide to form no, we'll take it out. I'll take it out. Then this cell produces outer primary parietal cells. So outer this cell divide and they produces the outer parietal cells like this. Outer parietal cells, and these are sporogenous, primary sporogenous cells. Here they produces these are called primary sporogenous cells. So what I have said, initially the anther remains too low and it is full of meristematic cells. This is meristematic cells. Some of the meristematic cells get dense and they have prominent nucleus. These cells are called archisporial cells. Then as the anther grow, the archisporial cells divide and forms an outer layer of primary parietal cells and an inner mass of primary sporogenous cells. Clear up to this? Now, these primary sporogenous cells these primary sporogenous cells will divide. Suppose this is a cell, this is a primary sporogenous cells. Inside it is there. These primary sporogenous cells divides periclinally. And anticlinally. What does it mean? What is periclinal and what is anticlinal? Let us go to the deep. Periclinally, suppose this is a cell, it divides from here. So there will be one cell here, there will be one cell here. Again it divides here, so there will be one cell here, and it also divides, there will be one cell here. So periclinal division will increase the length of the anther. So periclinal division will increase the length of the anther, right length. And anticlinally. So these cells divide periclinally and anticlinally. How? Anticlinal division, suppose this is a cell, it divides anticlinally. two cell. Again it divides anticlinally, another one cell, another one cell, another one cell, another one cell. So it will increase the circumference of the anther. It will increase the circumference of the anther, so it become more and more, circumference will be more, and the periclinal will increase the length of this anther. So this anther will look like this. So this will be flattened. So anticlinal it is circumference. So periclinal division will increase the length of the anther, and anticlinal division will increase the circumference of the anther. So anther will be, initially it will look like this. Initially it will look like this, and subsequently it will look like this. So it grows. The anther grows as it grows. So this produces layers. So many layers. So one layer, that is another layer. So here there are so many layers are being produced. Clear? So we should write periclinal and anticlinal. But in the exam hall, we will not be able to recollect what is that. I am giving you a simple tips. Periclinal is length. So I am writing PL. Anticlinal is circumference. I will write. Have you heard the name? This is PL account, profit and loss account. So in periclinal it will increase the length of the anther, and anticlinal it will increase the circumference of the anther. So if you remind PL account, you are through. Now this will give rise to so many layers, one layer, two layer, and three layer. So it will give multi-layered anther wall. Is multi-layered wall. So here it is now being protected. It is now being protected. It is so precious, the microspores, that the plant makes so many layers outside. So many layers outside. So here is one layer, two layer, three layer. So multi-layer protection gives the, this is primary porogenous cells from which spores or microspore will be produced. So it gets protected by producing this. This is being produced by primary parietal cells. Clear? I repeat. Initially, the anther is seen as a bilobed structure surrounded by epidermis and the inner mass of meristematic cells. First sentence. Second sentence you will write, as the anther grows, it becomes four-lobed. And some of the cells of this lobe, individual lobes, becomes distinct with distinct nucleus, and they become dense cytoplasm. Now these are called microsporangium. This is a microsporangium, 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 collectively called microsporangia, is microsporangia is plural. An individual is known as microsporangium. This is the microsporangium in which the microspores or pollen grains are produced, and they are protected by multi-layered anther wall. Multi-layered anther wall are produced by primary parietal cells. Clear? Now we will go to this cell. That is primary sporogenous cells. These primary sporogenous layer, they either these cells act as a microspore. Either they act as a microspore, or they may divide. They may divide and re-divide to form so many microspores inside. So these primary porogenous cells can act as a microspore or spore mother cells. They can act as a spore mother cell, or they can divide and re-divide to form numerous spore mother cells. So this is the microspores actually. Now, this I am taking it out. Either it acts as a spore mother cell, or many cells can be formed and which individually functions as spore mother cell. Now I am taking the spore mother cell, a single spore mother cell here. The spore mother cell divide and re-divide to form this divide. This I am making a little bit smaller. This is spore mother cell. Here, these undergoes two successive divisions. These spore mother cell undergoes two successive divisions. The first division is meiosis, and this division is mitosis. Very important. So, this is 2M. And the spore mother cell undergoes two successive divisions, of which the first one is reduction division or meiosis, followed by mitosis, and produces 4 microspores. They produces 4 microspores or pollen grains. Microspores are pollen grains. So microspore or pollen grains has been produced. But this is not gamete, mind it. These are haploid. Initially, they are surrounded by cell wall. The four microspore or pollen grains initially remains in bounded by a cell wall. This is called tetrad condition. Clear? So these are called tetrad conditions. These are all N. Clear? So these are called microspore or pollen grains. Now, the further they got separated and cell wall is formed. Around them. Finally, they got separated and this is being produced in microsporangium. Clear? This is being produced in microsporangium. Now, as these spore mother cells are growing, they should have food. So, on the other hand, I should say that the adjacent layer above the sporogenous cells, the adjacent layer is this. Say for example this adjacent layer. This is adjacent layer, adjacent to multi-walled or multi-layered anther wall. This is anther wall. These 3 or 4 are called anther wall. On the other, after writing this, you should write the anther wall, the innermost layer of the anther wall is known as, very important question, very important it comes. This is called EAPETUM. Tapetum, or I should say tapetum. So this layer above the sporogenous cells is known as tapetum. They are nutritive, Nutritive. They are nutritive in functions. They supply nutrition to the spore mother cell. So the spore mother cell get nutrition from the innermost layer of the anther wall, which is multi-layered, formed from primary parietal cells. This cell encircles the sporogenous tissue or sporogenous cells, or you can say pollen grains, supply nutrition. It is like you have seen that in a washing machine, you have seen that one container moves here and there, and outside there is a big container. So it moves, the inner container moves. So this is the inner container and this is the outer side. It supplies nutrition, and in case of washing machine, it supplies the washing powder. So it is known as tapetum, Tapetum. Very important question sometimes comes in the exam. Now, These 4 haploid nuclei are produced. These are 4 haploid nuclei. 4 haploid nuclei. 4 haploid nuclei are produced, covered by cell wall, and they act as pollen grains or microspores. Clear? Now the microspore will produce 2 cells. Now the microspore will produce, each microspore will produce by division 2 cells. It will be great. It will produce 2 cells, a large vegetative cell. So it is not large, that much large. So I am making it a larger one. A large vegetative cell and a small generative cell. Now the 4 haploid nuclei each will produce one big vegetative cell and a small generative cell. So in this 2 cell stage, the pollen grains bursts. The pollen grain, this anther bursts, and this is being carried to, this is male, this is being carried to the stigma of the other plant. In this 2 cell stage, now it will grow to produce male gamete. Now it will go, when it will fall in the when this will fall here over the stigma, then this will produce, this will grow, this will generate, which I am coming later. This will fall in the stigma of the ovary and they will penetrate here and they will fertilize the egg or ovum here. That is female gametophyte. Now this is male, and this male in the 2 cell, it is the anther wall bursts and it falls on the stigma by animals, by flies, by wind. So many are there. So that is the dispersal of fluid. From the dispersal of anther pollen grains, the pollen grains falls and then they will grow. That I will come in the next class. Next class we will generate this and we will start reading this. This is female gametophyte which will produce egg. Till then, take rest. Thank you very much.